Muy buenos días. Mi nombre es Ernesto Gosser, soy médico, profesor de la Universidad Peruana Cayetano Heredia y les doy la bienvenida a este nuevo webinar que va a tocar un tema muy importante que tiene que ver tanto, bueno, ya va a ser presentado por el doctor Peter Gámez y después haremos una conversación. Este webinar va a ser un webinar en inglés. Entonces, en este canal, donde me están escuchando, todo va a ser en inglés. Y si ustedes se fijan, en su pantalla, en la parte inferior, a la mano derecha, hay un símbolo como de mundo que dice interpretación. En ese símbolo, ustedes pueden, los que quieran escuchar en castellano, hacen un clic en español. ¿Ya? Como en los canales de streaming, ustedes escogen el idioma en el que quieren escuchar. Si quieren escuchar en inglés, pueden permanecer en este canal. La doctora va a hablar en inglés, el doctor Gámez y yo vamos a hacer comentarios y preguntas en ese mismo idioma. Entonces, ya sabemos las cuestiones técnicas, ahora quería informarles a todos los que ya están inscritos en el diplomado y a quienes estén interesados todavía, que ya se aprobó el diplomado ya tiene la cantidad de participantes que la escuela de posgrado necesita para aprobar el inicio de un diploma, y va a iniciar sus clases el día 26. Hay varias personas que nos han solicitado una inscripción extemporánea, entonces hemos ampliado para esas personas que todavía no tienen sus documentos completos hasta el día, me parece, 12 de marzo, la inscripción. Entonces, para que completen sus documentos, porque como ustedes saben, se necesita tener los documentos completos para poder ser evaluados y ser aceptados o no en el diploma. El diploma va a tener profesores invitados nacionales y extranjeros de la calidad como la invitada de hoy día, que va a ser presentada por el doctor Gámez. Entonces, vamos a dar inicio a esta sesión y ahora invito al doctor Gámez Dr. Gámez, please, can you introduce the topic and our guest today? Go ahead, please.
Thank you, Dr. Gosser. Today we are going to talk a very important theme in science. Nowadays in Peru, we are living a war against a very difficult and weird disease. When this starts in our country, I was in first line fight in my hospital, and the knowledge that we had on that time, it was really short, and many patients died in that time. Today, we understand more about COVID-19, and we have more tools to fight against this disease. I studied cannabis many years ago, so it was really interesting to me to find information and some studies that show some potential to fight or to treat this disease. We know that the inflammation that this virus causes in our body, even when it's not in our system, it's really heavy. So cannabis or many cannabis profiles has the potential to treat inflammation. Even not only COVID-19, even in other diseases like lupus or arthritis. But this day, I want to introduce the professor Hinanit Koltai. She is a PhD. She works in agricultural research organization at Volcanic Institute in Israel. We know, all of us, we know that Israel is the top, has the top of investigation in the world, and she has a published article and at Nature, and this year was published, and it was titled "Cannabis Compounds Exhibit Anti-Inflammatory Activity in Vitro in COVID-19 Related Inflammation in Lung Epithelial Cells and Pro-Inflammatory Activity in Macrophages." Good morning, Professor.
Thank you very much, Dr. Gomez and Dr. Gozer. I'm very happy to be here virtually and to tell you a little bit about the studies that we do. And for that, first, I will share the screen. And then, indeed, the topic is, as Dr. Gomez presented, but to fully understand our approach, I thought to start a little bit earlier than this research. And indeed, as Dr. Gomez mentioned, and I understood that and I fully support what he said, is that we started from the plant. We started from medicinal plant and cannabis in particular. And what we aim in the last few years in my lab, we aim to decipher and understand what compounds and what composition of compounds are active in medicinal plants in general and cannabis in particular for different medical conditions. A few words about myself. I'm a scientist in a Volcanic Center, which is a governmental institute for agricultural research. I'm a scientist there for 20 years now. And during these years, I worked on different subjects in my lab. We worked on plant hormones, but in the last 6 years or so, we are working on medicinal plants and cannabis in particular. So indeed, let's start with cannabis. And just briefly, cannabis is very well known as a medicinal plant. We have no doubt that cannabis is highly helpful for the treatment of different medical conditions. It is not healing, but rather mainly alleviating symptoms that are associated with pain, with inflammation, and additional ones. Cannabis is a herbal plant, flowering plant, and it contains only one species, which is Cannabis sativa. The other names for cannabis, like sativa and indica and wooderalis, are merely sub-species to Cannabis sativa. The plant is dioecious. It leads to the formation of male plants and female plants. And the flowers that develop on male, those that may fertilize the female flowers to form seeds. However, unfertilized female flowers produce small hairs like on the flower, and these contain secondary metabolites that are being produced in these trichomes on the leaf and are being accumulated in the trichome in such a way that the female flower is completely full of these small hairs, as you can see here. And these trichomes are the ones that contain the compound.
Yo no veo que ninguno esté en español.
Cannabis contains hundreds of different compounds, hundreds. It contains about 150 phytocannabinoids, which are the compounds that are specific to cannabis. Contains hundreds of flavonoids, hundreds of terpenes. So it's really rich in compounds, this plant. Just to say that flavonoids and terpenes you can find in other plants, but not phytocannabinoids, which are unique to cannabis. I'm not going to say much about these compounds, but this is one of the references that you can have a look at and see about phytocannabinoids. Just to say the THC, which is one of the phytocannabinoids, is the only psychoactive compound produced by cannabis, and that the plant produces the acid form of these phytocannabinoids compounds. And just to say that Professor Meshulam from Israel and co-workers identified the major phytocannabinoids back in the 60s. Another phenomena that we should take into consideration is the entourage effect. The entourage effect is based first on some evidences in the literature, as this one, for example, but also on the reports of patients that say that using the whole flower, which contains many different compounds, and just to say that in each flower that we use, there are several dozens of compounds. This is much helpful than using one compound only. This became to be called the entourage effect, and suggests that combination of compounds from cannabis are better than using only one compound. We'll get back to that in a minute. Okay, but despite the fact that we know that there are many compounds for cannabis, still the medical use is based on non-scientific terms. First, it is based on using terms like sativa indica or duralis, and I told you that these are not different species of the plant, but rather subspecies, which are inbred again and again. So definitely, and we'll talk about that in a minute, that's not the way to go. Definitely not putting some terms to be used for the medical use, like mind dominant or body dominant. Or these are not medical terms. I don't have to tell you that. You know much better than me, but patients should be taken care in medical tools. And then in the best case scenario, cannabis is referred to, cannabis products are referred to as THC or CBD. CBD, another phytocannabinoid that is good for the reduction of inflammation, for example. And that's it. So products or the flower itself are only characterized in the best case scenario by these two compounds. And we know that there are hundreds of compounds and dozens of compounds in each flower that we take. So definitely, we do not see here the full picture. On top of that, there are hundreds of different cannabis strains that are merely inbred lines that were bred for hundreds of years. We'll talk about that in a minute. And in such a way that we have today hundreds of different strains. Nobody knows what what compounds are present in each of the strains. There's a lot of mythos and stories about the ability of these different strains. And you as physicians, especially when you want to go and treat your patients, how are you going to treat them with cannabis? Are you going to treat them with what? With Genesis? With revelation? Definitely, this is not medical treatment. And this is basically our purpose, to take cannabis into the medical world and to put in your hands, physicians and MDs, to put in your hands the ability to treat patients with cannabis in a proper way. And again, this is something we'll talk a lot in the upcoming minutes. So about the strains, indeed, there is no clear definition of the concept of a cannabis strain. This is definitely also in the scientific literature. Just a brief history of cannabis. Cannabis, both a subspecies sativa and indica, are originated from the Himalaya, from India, in the upper grounds of the north of India. The plant was distributed first to the east and then to the west by men mostly, and we know that based on archaeological evidence that suggests that cannabis is being used by men more than 6,000 years. This is based on evidences of seeds and also of fabric, pollens, fabric as well, and you'll see how this is connected in a minute. If we are looking at cannabis today with the hundreds of different strains, based on genetic tools, we are able to define several different lineages that led to the development of the use of cannabis in different parts of the world throughout the years. And this is because of breeding and selection by men for cannabis that may fulfill the purpose that men wanted it to fulfill. For example, in Afghanistan and Pakistan, for example, cannabis was grown to produce hashish. Hashish is basically the trichome, the parts that contain the active compounds condensed together, and these have very high level of active compounds, including THC, definitely an illegal substance and not used for medical use. This is for hashish. In other parts of the world, cannabis was bred for marijuana, for example. And marijuana, the dried flowers, female flowers of the plant that are the source for the active compounds. This was done, for example, in South Asia, in Africa, and so on. And in other parts of the world, cannabis was bred for a totally different purpose. And this is for fiber and seeds. This plant, this was mainly in Russia and the Mediterranean, and these plants grow high. They have very strong fibers and are being used for fabric. And as I said, fabrics, the canvas fabrics are the ones that are composed from hemp plants, which are cannabis sativa, bred for this purpose. The composition of compound is in accordance to the purpose that men wanted to have from the plant. So for example, for hemp, you have very low amount of cannabis compounds, and you have different compositions as needed for the other uses. So again, if we think of the strains, we can divide them to these different lineages developed by men throughout the thousands of years that man is using cannabis. If we look at the population of cannabis strains, we have wild plants that are grown in the Himalaya, as said. We have land races that man started to grow and they escaped to nature at some point during history. And we have the inbred lines. And these are the ones that are used for medical cannabis. Israel is going through a process of medicalization. And in this process, we are trying to close the gap that we have between the different strains and compounds and the non-professional and non-medical use that is being done with cannabis. And we try to close the gap to give physicians tool to treat patients in a proper way. And you can see here, just as an example, I know you cannot read everything that is written here, but this is in Hebrew, of course, the table that is being provided today to MDs in Israel for treatment of patients. It's not perfect yet, and it is still based on THC and CBD, but still it's at least trying to move towards a proper medical treatment. Okay, so indeed, to close this gap, many have done many things. I will tell you about what we have done in collaboration with the Israeli Gene Bank. What we've done is we established a large collection of cannabis seeds. The Israeli Gene Bank was able to import from different gene banks throughout the world to import different cannabis strains and to characterize these strains. You can see here, just for example, a small example of the different strains that the Israeli Gene Bank has. And for example, this, as you can understand, is hemp, because it has very long fibers which do not branch, and very small flowers. This is, for example, marijuana, because it is growing short, has many branches and flowers, many flowers. So this is just an example. The Gene Bank characterized the collection of seeds based on markers, genetic markers, which are single nucleotide based on single nucleotide polymorphism, but I'm not going to go into deep here. And just to say that based on the public knowledge on cannabis genome, we were able to characterize each and every line that we have, each of the interesting or successful lines that we have in the gene bank, based on these single nucleotide polymorphism differences. So this is, again, I don't want to go into too many details here because we have a lot to talk today, but this is basically fingerprinting of the different lines based on genetic profile. We've also characterized the different compounds produced by the different strains in the gene bank. And another part of the activity that we do, not me myself in my lab, but other labs in my institute and in other institutes, we are working on generating good agricultural practices to grow cannabis the best we can. Okay, but as said, I think cannabis-based medicine should be evidence-based medicine. And I think this is the biggest challenge that we have today to take cannabis and to push it into the medical world in a way that will give you tools for the treatment of patients. And this is the subject that my lab is working on. Just before continuing to telling you what we do in my lab, I want to tell you a little bit about the answer to the question. Is what is the basis for the profound effect of cannabis on our body? Why cannabis is so effective, and we do know that it is effective. And again, here I'm not going to talk into too many details, but I have to mention that part of the findings of Professor Meshulam and co-workers was that cannabis compounds attached to the endocannabinoid receptors, these are binding THC in particular and CBD to some extent. And these receptors, CB1 and CB2 and additional ones, are dispersed throughout the body. CB1 is dominant in the central nervous system. CB2 is dominant in the immune system, but basically they are dispersed throughout different tissues and organs in the body. This includes the muscles and lungs and the colon and so on. I think the breakthrough, perhaps, that Professor Meshulam and others did is by the understanding that these receptors, the endocannabinoid receptors, indeed they bind THC and CBD to some extent, but this is not their purpose. It's not that we hold receptors to bind molecules from plants. We do not. But rather, these receptors are binding endocannabinoids, which are endogenous ligands that are based on the fatty acids. These endocannabinoids are the natural ligands of the CB1 and CB2 receptors. And they're the ones that with high affinity bind these receptors and regulate so many different processes in our body. Again, of course, I'm not going to talk too deep and not too much about that, but just to give you a glimpse into the importance of this system is one, for example, they regulate endocannabinoids and the receptors regulate the level of neurotransmission and the level of release of neurotransmitters into the synapse between two nerve cells. So this is one very important activity, and by that, the endocannabinoids throughout the receptors, especially CB1, attenuate and regulate the release of different neurotransmitters. Now you can understand how cannabis and THC in particular is affecting the central nervous system in this way. And this is by binding to CB1 in this case and leading to inhibition of release of neurotransmitters. This definitely explains at least part of the activity of THC and CBD as well. And then it leads to an understanding of the profound effect of these compounds on our body. I want to mention one more phenomena, and really I suggest to read this wonderful review by Professor Maccaroni from Italy, because it really explains the involvement of phytocannabinoids and endocannabinoids in the nervous system. Just to say that it turns out that in a developing mind, like those of children and adolescents and young adults, the nervous system is being still developing, still generate, and the nerve cells are migrating throughout the brain into their position for the further development and differentiation. And it turns out that both these process of migration and differentiation and development of these cells is regulated by endocannabinoids and the CB1 receptor. So again, on the one hand, it really demonstrates the importance of this system and the effect, the profound effect of phytocannabinoids. But then it also says something about an uncontrolled use of cannabis. And it really, I believe, should make us worry about children, adolescents, and young adults using cannabis and interfering with these so carefully regulated processes. Okay, so this was just to give you a notion as to the mode of action of these compounds, at least partially, but we will talk about that a little bit more in a minute. And now I would like to take you into the work that we do in my lab. We talked about that definitely medical cannabis provides symptom relief for patients for different medical indications. However, there are about 500 different compounds. So we asked from the beginning of our work, what compounds are the active ones? And it turned out that 6 years ago, when we started to work on cannabis, there was very little amount of information on the activity of the individual compounds. Mainly THC and CBD were known in terms of activity, and some of the others as well. There was some information on the use of the whole plant, the whole flower, this way or the other, but there was no information about the combination of compounds that are important for the treatment and may give optimal results. And this is so important because of the entourage effect that we talked about earlier in my talk. Just to remind you that this entourage effect suggests that several compounds together are working better than a single compound only. So we ask not only what compounds are important, but rather we ask what composition of compounds from cannabis are the best ones for the treatment of different medical conditions. And this is something that was totally unknown when we started the work. Okay, so to answer that and to determine the optimal composition of compounds from cannabis for different medical indications, first we have a very small greenhouse near the lab where we grow some cannabis plants and we take the flowers. We also get flowers from growers. And all these are giving us the ability to do extraction and to get the different compounds. We use some regular, I would say, extraction protocols. And then we combined two disciplinaries. One is chemical analysis and the other one is biological analysis. And this is how we do it. After extraction, we separate the extraction to fractions. And then in each and every fraction, we can do chemical analysis and see up to the last molecule that we have there. And each of the fractions is subject to bioassays, to biological assays, to isolate, to identify the active fraction. Once an active fraction or fractions are identified, they of course go through a fully chemical analysis and additional biological assays to determine, for example, the mode of action. For each and every medical condition that we work on, we form collaborations with physicians in Israel, in different hospitals. And with them, we build the best assays that we can, which are based on in vitro, ex vivo, getting from the hospital under Helsinki approval, getting different tissues, biopsies, blood cells, as I will show you in a minute. And we also started to print tissue in my lab and work on them to better represent as much as possible the disease. This collaboration with physicians from hospitals in Israel allow us to go to clinical trials, and then new products are developed based on this research. New products are developed by companies, commercial companies. I will show you two examples. One would be on a certain skin disease, and the last one would be on COVID-19. Okay, but let's start with the skin. Both mycosis fungoides and Sézary syndrome are stages of cutaneous T-cell lymphoma, CTCL. The mycosis fungoides is the most common type of CTCL and is apparent as patches of malignant T cells in the skin. Sézary syndrome is the leukemic type of CTCL where the malignant T cells are already dispersed in the blood, and then prognosis is not a favorable usually. Just to say that this is a close collaboration with Beilinson Hospital in Israel, with dermatology department there, and the idea to work on this subject with cannabis came from the physicians, from the specialists themselves. They, when we started to work on some other medical indication related to skin, they came and say, listen, patients, CTCL patients, that we've tried everything and we've nothing to give them anymore. They are resistant to the different treatments used. When they take cannabis, when they get cannabis, who knows from where, doesn't matter, and they put on the skin, the situation is getting much, much better. So they said, let's work together and find what cannabis compounds are or compositions are the ones that give this effect. So definitely this is what we started to do in this collaboration. And as you can understand, when you have 500 different compounds and different strains, and you need to find combinations, then you have many, many different possible matrices that you need to check. And this is definitely a challenge, but a challenge that we learned how to do in a reasonable time and efforts, and how to find compositions that are highly effective. We do that using the pool assays, where we take a pool, several pools, and if one of them is active, then we divide this pool to sub-pools. We again check for the activity, and then again taking the active sub-pool and again fractionate it to sub-pools, and so on, And in this way, we are able to screen many different combinations and to find the most active ones. So as said, we have extracted compounds from different cannabis strains. We have divided this extraction to different fractions. And in this approach, we were able to identify two fractions, S4 and S5 here, that are highly active against the malignant T cells. But also we found that if we combine these fractions together, then we have activity which is 20-fold higher than the one of each separately. So it means that if we take 1 and 1, it's not 2, it's 20. And this is because of synergistic interaction between these compounds. This, on the one hand, leads to the ability to use very low concentrations to treat cells and possibly patients with relatively low dosage, which is very important. But it also leads to the ability to better understand the entourage effect. I told you about the entourage. I told you about that this is definitely something that patients report, but the basis of the entourage was not clear. We think that this synergistic interaction between compounds is the basis for the entourage effect. This is all published in OncoTarget last year. Okay, so as I said, we identified active fractions, we saw synergy, and we were able to identify the composition of the different fractions, and this is just an example. And this composition contained in certain ratios, a percentage, different cannabis compounds. For example, here CBG, CBN, CBCA, and so on. This we were able to protect by issuing a patent, a composition patent on this composition. And then this was could serve as the base for the development of new cannabis-based products. I'll get to that in a minute. However, we were very much intrigued with this synergy between the compounds. And to better understand the synergy, which is so intriguing, we exposed the cells to human cells, of course, of the first of cell lines, and then of patients of the malignant T cells, and looked, for example, on apoptosis. We were able to see that only the synergistic combination is leading to cell apoptosis, programmed cell death. Then we sequenced the RNA being produced by the different cells treated with one fraction, another other fraction, and with the synergistic combination. And in 2 cell lines, we were able to see that around 3,000 genes are being overexpressed or repressed due to the synergistic interaction. This led us to the understanding that new biological events are happening once the cells see the synergistic mixture of compounds. Then we were able to identify some of the signaling pathways that are involved, that are being activated or repressed in this synergistic reaction of cells to the cannabis compounds. And this is just an example of one of the one of the signaling pathways, which is basically a target for inhibition by several chemotherapy drugs being used also for CTCL. So just to say that this leads us to the ability to talk about cannabis, not only on the composition of compounds, but also on the targets that these compounds are working on, the targets that they are affecting. This is a whole new nomenclature for cannabis. But the most exciting, I think, result was the one that we got on cells that we've received from patients' blood lymphocytes, peripheral blood lymphocytes of Sézary patients. There, with the hospital, we were able to see that first we can induce indeed apoptosis, as said, in these PBL of patients, and then that we're able to affect PBL of malignant T cells at a higher level, significantly higher level than non-malignant T cells of these patients. So definitely this paved the way to clinical trials that I hope that we will commence very soon. But then when this awful pandemic started, we felt that we need to do our best and at least give something to humanity, some clues. And we knew that cannabis is able to reduce inflammation. We have several projects that were able to show that, and others, of course, showed that, not just us. I've not shown here, but this is part of our publications. And we asked ourselves, maybe cannabis, maybe it could be used for the treatment of COVID-19 patients, and especially the related acute inflammation that is associated with this disease, because cannabis is known to be immunomodulator and anti-inflammatory. Just a very brief introduction. I'm sure all of you know very well about this disease, no doubt, better than me probably. But I would like just to say that, to remind you that this disease is, we can see two stages in this disease. One is one that requires a specific adaptive immune response, and this is to eliminate the virus particles and prevent disease progression. And the other one, the one that is more dangerous, the one that is lethal, is the cytokine storm syndrome that is developed in those that are severe patients of COVID-19. And this is associated with a very dramatic induction in interleukin 6 and 8 and inflammation of different parts of the body, lung, but also other parts of the body that are getting highly inflamed. And of course, this is a very dangerous situation. Again, I don't think I have to tell you much about that, but briefly to remind you that the lung is covered with epithelial cell, and this is especially in the alveoli, which this is where the gas exchange take place. And then in these alveoli, in addition to the epithelial cells, there are many different cells of the immune system, and in particular macrophages, which are the ones that involve with really the eradication of virus and bacteria and other dead cell different particles from the lungs and from other parts of the body. These macrophages move very smoothly between the lungs and the blood and different organs of the body. And it turns out that in this disease, they're the ones that carry with them inflammation in the lung and to the different parts of the body. So we worked on these two cell types and we asked, may cannabis be used for reduction of the COVID-19 related acute inflammation? And as said, this is just published just a few weeks ago. And what we found is that indeed certain strains of cannabis are able to reduce inflammation. And you can see here, just as an example, this is in epithelial cells of the lung, where inflammation is being induced by TNF-alpha. Our positive control is dexamethasone for reduction of inflammation. And then one of the cannabis strains were able to quite nicely reduce inflammation in these cells. And when we looked on interleukin-6 as a marker, as a very good marker of inflammation. Once we fractionated, as I told you before, we fractionated the extract of this strain, we were able to get one fraction that is more active than the crude extract and another fraction which was less active and we did not continue with it. This was rich in THC. The one that was highly active was rich in CBD. It was definitely not just CBD, as you will see in a minute, but it was rich with CBD. Okay, so this looked very good and the specific activity was high. So we were very happy with that. However, when we started to work on macrophages, it turned out surprisingly that cannabis, the crude extract, and the fraction itself are leading to high induction of inflammation in macrophages. And this is definitely something that is really worrying in this respect because it may worsen the cytokine storm. We could see that in, for example, a 400-fold induction of IL-8 and 2 induction of IL-6, and this is above and beyond what TNF-alpha even could do, or other inflammation factors. We could also see that by the change in formation of macrophages and the polarization of these cells. You can see here one macrophage, just as an example, of course, one macrophage which is mature, but it is not activated. And this is in the control. And look what happens when we add cannabis and the fraction to these cells. They become polarized and very active in terms of the secretion of the interleukins, but also in terms of activity of phagocytosis, of collecting particles. And here you have nanoparticles stained in green that you can see that in the control, this macrophage cell is quite reluctant to collect them. However, when we add cannabis, definitely these cells fully collect as much as they can these particles. Basically, this might be a very good activity in some instances. And in some diseases that suffer from low level of the macrophage activity, this might be very interesting. Definitely, this is something we're looking into now. But this is not something we want to have for the treatment of COVID-19 patients that already suffer from highly active macrophages that bear with them inflammation throughout the body. So, first of all, we definitely said in this paper that cannabis as is cannot be used for the treatment of COVID-19 patients. It may worsen the cytokine storm. And this was very important for us to say, especially because of publications, not scientific, but rather media publications that said that cannabis may be used, and it was like published by different commercial entities that, oh, we have cannabis that can be used for COVID-19. And this finding was really alarming, and we had to share that. That's one. But then in the next stage, we asked ourselves how we can improve the activity of cannabis. And what we found is that when we use only the compounds that are present in the only these compounds as they are present in the active fraction, the fraction that was active on epithelial cells of the lung, then the system looks different. And indeed, we still can reduce inflammation in epithelial cells, and we do not ignite these macrophages to the extent that the fraction is doing. We show that in different ways, in the secretion of IL-8 and interleukin-6, and then by sorting cells that ingest, that got with phagocytosis different nanoparticles. And this was definitely something, just maybe a light at the end of the tunnel in this case of cannabis to be used for COVID-19. And this is again all in the paper. What we do now is we look for combination of cannabis compounds with different pharmaceutical drugs, not just to not induce inflammation in macrophages. This is not good enough. To fight the cytokine storm, what we need is to have high activity against inflammation also in macrophages. And definitely this is something that we look into now. And this is something I cannot publish yet, and I cannot say what compounds we are working on. This is true, just an example of some of our true results. And I think that this is a synergy map, and I think that you can just see that when you use the compounds by themselves, it's not good enough. This is a certain compound, I'm not going to say which one, but this is pharmaceutical drug, and when you use it by its own, it is not very active. But when you combine FCBD, not the fraction, but the active compounds, as I showed here, then you can get highly synergistic activity against inflammation. So definitely this is something we are doing now. And this is summarized. Okay, so just to summarize what I talked about today. We started from different types of weed or strains of cannabis, which are not defined. We went through a chemical analysis, bioassay, and identification of active fractions and active compounds. We identified synergies as well. We determined the formulation in terms of active plant ingredients from cannabis for different medical conditions. And just to say that we already know But different medical conditions demand different compositions of compounds. So we can specify the treatments and say that for certain medical conditions, a certain compositions are needed, sometimes more than one. We identified the targets and looked a little bit into the mode of action. In all cases, we also look at the receptors that are being involved. I have not talked about that much today, but definitely this is something that we do. And then with our collaborators from hospitals and with commercial companies, we, mainly the companies, are doing whatever tests are needed, like drug-drug interaction and toxicity. And this is all steps for the development of new cannabis-based products in different delivery methods. Those that would be not just based on some oil or some flower or joint, but rather those that will be given at some form, suppositories, for example, but this is just an example, of course, and this will be based on formulation of certain API from cannabis, which are the best ones for treatment of this medical condition and alleviation of this and other symptoms. This also leads to the development of elite chemovars of cannabis that will not be called White Widow or Genesis, but rather will have certain composition of compounds. As much as possible, of course, it will never be exact, but they can, to some extent at least, have composition of certain compounds that is needed for the treatment of medical condition. And in the future, I hope that we will be able to see cannabis strains that are designated for the use, for the treatment of certain medical conditions and will be called, for example, IBD1 strain or CTCL3 strain, and so on and so on. And with that, I would like to finish with acknowledging, first of all, people in my lab, which are so dedicated and so excited to do the different projects that we do. The Israeli Gene Bank and the head, Dr. Einav, the head of the Gene Bank, and then our collaborators from the hospital. And today I talked mainly on Beilinson Rabin Medical Center and the collaborators under Professor Emilia Hall. And with that, I would like to thank you.
Thank you very much, Dr. Colte. It was very interesting to listen to your presentation because you started by providing the background and then going with examples of other uses similar to what you are studying, and then more in deep to what you are studying. And I want to start by emphasizing what you said, because has been a problem here in Peru, that you underscore the importance of evidence-based medicine. And I want to underscore this again by saying that we can't jump to conclusions. This is a very good study, and this is in the lab. And to go to the humans, we need to travel a road but is still ahead. So I also have a question. There is one question from one of the participants, because you are discussing about this the effect, the anti-inflammatory effect on on lung cells of the some composition of cannabis. And what do you think about could happen in the future when we go to the humans, and we will need to have a different type of compounds with different composition for different patients, or what is important, for instance, in the clinical trials to actually try with different type of people to really understand the effect on humans. Over.
Absolutely. You raised here a very important point, and this is that treatment may be considered only after clinical trials. This is very important. And then, as per your question, what we try to do is we try to test the compounds, or the different composition of compounds on as many models as we can. For example, on different cell lines, and if possible, on samples from patients. This allows us to check for effectivity on the different cells, especially when they come from patients. And then what we look for, indeed, are compounds that are working on the different cells that we get also from different patients. For example, with the CTCL, just an example, we worked on many different cells, peripheral blood lymphocytes from patients and from normal individuals. And by that, we were able to determine what are the best compositions that may be active on the patients. And we looked for those compositions that are active on many of the samples that we checked. Again to say, so this is still ex vivo and in vitro. This is not clinical. What the physicians that we're working with are doing is they are taking these results as evidence-based medicine. And then what they do is they translate this information to clinical trials. Only after successful clinical trials we can say that we have developed something that could be used in patients.
Thank you. Dr. Gomez, do you have a comment?
Professor Finani, thank you for the presentation. We all know a lot about endocannabinoid system. It's obvious that endocannabinoid system is altered by COVID-19. So I don't know if you explored that, but have you seen a change in the number of receptors in the lung?
What in this case of COVID-19, what we did indeed is we looked at the receptors that have been involved in this activity. And we found that this activity is mediated via CB2 receptor mainly. And then what we did is we determined the level of CB2 receptor in these cells. Unfortunately, here we could not compare between different patients because of biosafety, but we were definitely able to see a relatively high expression of CB2 in the epithelial cells. It is hard for me to give you a full answer because in this case, we could not examine biopsies. Of course, it's not that available. And it's not the, it is a biohazard problem.
Okay.
Yeah, so I cannot answer fully in this case.
Yes, okay. I can see that in the study, the entourage effect concept is reinforced. Oh. For future, what do you have in mind to study related COVID-19 and cannabis?
What again? Could you ask again? What is?
What studies do you have in mind to make with cannabis and COVID-19?
Okay, good. Yes. So what we do now, indeed, is we look for combinations of the cannabis compounds and pharmaceutical drugs. We have a collaboration now with a specialist, lung specialist from Sheba Hospital in Israel, Sheba Medical Center. And what we consulted with him is about the different pharmaceutical drugs that can be used. We know now, we know today that most pharmaceutical drugs being used, most of them are not that effective and not for everybody. We're looking for the combinations of the cannabis compounds and these drugs to be able to really reduce inflammation in macrophages. We found that reducing inflammation in different cell types and the epithelial cells is much easier than reducing the inflammation in macrophages, even with the pharmaceutical drugs. So the first thing is to find the ability of cocktails to be used for effective treatment. Once we will find these cocktails with the specialists that we're working with, we will go deeper into the mode of action, and I hope in some point with him, we will be able to start clinical trials.
In some hospitals around the world, they are reporting some interesting results. I am not sure that is the way to use cannabis, but how about the clinical observations about the reduction of symptoms of COVID patients that some clinicians are reporting? Is that a clue?
With cannabis, you ask?
Yes.
Yes, yes. I tell you, the problem that we suffer with cannabis is that nobody knows, not just with COVID-19, but also in general, nobody knows what the patients are really taking. This is because the cannabis is not well characterized. And when the patients take cannabis, not them, not the physician, nobody knows what really is working, what they're taking, and what is working among the different dozens of compounds that they're taking together. This leads to a problem of standardization of medical care. It means that it's very hard to keep treatment in a high level because different lots, for example, give different effects. And the flowers from this batch are different from flowers from the other batch. And since nobody knows what is working, this work of defining the active compounds, I believe, is much needed. So cannabis definitely has a potential, and this is why we're working on it. But without defining the active compounds, there is not much ability to provide proper medical care with cannabis.
Thank you for that answer. And I repeat this because this concept, these words have to be clear, for clinicians that can see and listen us. Dr. Gosser?
Okay. Dr. Koltai, one of the participants asked which terpenes or combination of terpenes in general have a more potent effect, anti-inflammatory effect in general, not only related with your study, but in general, which of them are the best?
We asked ourselves whether terpenes are important at all, because we know that phytocannabinoids bind receptors, and there were quite a number of studies that suggested that terpenes are not part of the entourage effect. We asked ourselves whether terpenes participate at all in the activity of cannabis. What we did is in a published study, I think it was 2 years ago or 1 year ago, I don't remember. What we did is we looked for the distribution of terpenes in 70 different lines of cannabis from the Israeli gene bank. We found that terpenes are correlated in the appearance to the appearance of certain phytocannabinoids. So it means that the distribution of terpenes is not random, but rather they are grouped into groups that appear with a certain phytocannabinoid. Then we asked about the activity. We asked whether phytocannabinoids that come with THC are working with THC. And we found that they indeed, in some of the cases, they induce activity. More than that, when we took terpenes of THC plant and mixed them with CBD, we did not see this induction of activity. So it means that the entourage effect of terpenes with phytocannabinoids exists, and that it's specific. It's not any terpene with phytocannabinoids. Now, if you ask what terpenes, this we do not know. We know that there is a group that contains quite a number of dozens of terpenes is working with phytocannabinoids. We still do not know what terpenes in particular are working.
Mm-hmm.
That's to say that this again change between different medical conditions. Mm-hmm. Because in some of the cases, we don't need the terpenes. The phytocannabinoids do all the work. In some cases, we do need.
Okay, thank you very much. It was very, very useful. And as you know, there is also in COVID, coming back to COVID, and there is a long haul cases where the people remains with the disease, and it is mainly due to inflammatory effect of the virus in the body. I have seen several patients that several weeks or even months remain with some difficulties to breathe and this and that. Do you think there is a place for cannabis there, and how can we accelerate the studies to help that? Because we know that there will be millions of people with post-COVID, and I have seen even mild cases that after recovery come with this small but burdensome problems with in recovery. Do you think? My question is: do you think is there a role for cannabis, and how can we accelerate the research that we need to help them? Because, thank you.
To my understanding, cannabis is definitely can help in reducing inflammation. This way or the other, it can be used to reduce inflammation, but again, you should be very careful with it because in some cases it may even induce inflammation. So the point is that you have to use only certain combination of compounds for this reduction of inflammation. Whether this can help also in the post-corona symptoms, I do not know. I do not know because first of all, it was not tried, and then maybe we do not know enough on the post-corona symptoms, on the origin of these symptoms. So I would say that if this is all a matter of inflammation, maybe certain compounds from cannabis may be used. But again, if it's something more, if this relates to other physical conditions, then everything should be tested. I do not know. As per your question, is how you can speed the research in this regard is, I would say, from my side, is to start conducting clinical trials. To do it very carefully, to start with toxicity studies perhaps, but not to waste any time, but rather to start clinical trials. And I just say that I'm very open for collaborations. And if anybody is interested in translating our results as possible to clinical trials under authorization, of course, of what is needed in your place, then definitely this is something that I would be very happy to collaborate.
Mm-hmm. And one question related with that, because I have heard this in several places. What is your view, not your, on this affirmation that the cannabis could prevent coronavirus infection? I would like to hear you.
It cannot. It cannot prevent. No way. No. I heard that too, and I don't think so. It cannot prevent a coronavirus infection, but rather, this is how I see it, how I think, but rather just reduce perhaps the level of inflammation.
Mm-hmm.
Definitely, there is no replacement for immunization.
Okay.
No replacement for immunization.
Thank you very much, because this is something that people is in these times, not only in Peru but in other countries, we are trying to find the magic bullet, and we are founding many magic bullets, and this is a very dangerous to me to think that that if this is anti-inflammatory, then it will protect you. If you are being very clear that it is, because the answer to that question is no, just that. Thank you very much. Dr. Gámez?
Yes, I think that we are closing this webinar. Again, thank you. Again, Professor Koltai. I know that this was really fast, and maybe we need more time. And thank you again. Thank you because your work. And I hope that this is the beginning of a professional relationship with Dr. Gosser also.
Okay.
Very good.
Thank you very much, Dr. Koltai. On behalf of the university, this is Cayetano Heredia University, and we are starting the first certificate on medical use of cannabis, and we are very thrilled because we are about to start this certificate to prepare the people to use the evidence to treat patients and not to put them in danger. And I really appreciate your kindness of accepting this invitation, even though we didn't meet before. It's usual that you invite people you know, and they are very happy to do that. And so I really appreciate that you spend this time with us and in line. And to me, I want to underscore another thing, that this is the work that you are doing in Israel, you and other groups, it is very important. Research is the foundations for making real good decisions in public health and in medicine. We need to do research, basic research, clinical research, public health research, to be sure that what we are doing is something that is in benefit of the humankind. Thank you very much, Dr. It was a very pleasure.
Thank you so much. It was my pleasure. I was very happy to share with you what we do and what we know. So thank you for your time.
Okay, thank you very much. Goodbye.
Bye-bye.
Thank you to all the participants of this webinar that has been the first webinar in Peru done in English and in Spanish, with the Spanish translation at the same time. Thank you to Dr. Gonzalez for the translation. I have heard that it for some time, and it's very good. Thank you very much to all the team, because there is a team behind. And I also want to thank our partner, Anden Naturals, that is helping us with this to the university in this webinars. Thank you very much. Bye-bye.
Thank you. Bye-bye.
Thank you. Good afternoon.